Hello, my name is Soraya Smith, and today I will be presenting a publication titled CRISPR-Cas9 mediated genome editing corrects dystrophin mutation in skeletal muscle stem cells in a mouse model of muscle dystrophy. The authors' names are listed as a reference. So what even is CRISPR-Cas9? Well, CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, and Cas9 is the protein that helps catalyze the process of cutting out specific segments of DNA. In class, we learned that proteins come in various forms and can perform a variety of functions. So, we can think of CRISPR-Cas9 as a gene editing system that is made up of an active protein complex, and this complex can basically remove undesirable segments of DNA to fix things like genetic mutations. Duchenne muscular dystrophy is an X-linked recessive genetic disorder that causes muscles to degenerate. Any muscles in the body can be affected by this mutation, including skeletal, cardiac, and respiratory muscles like the diaphragm. Individuals with this genetic disorder face developmental barriers and shortened lifespans. The causal question for this experiment was, can CRISPR-Cas9 gene editing enhance correction and/or prevent the DMD mutation? The hypothesis was that CRISPR-Cas9 would increase expression of the muscle fiber protein known as dystrophin. Based on the publication, the prediction was if human intestinal cells instead of embryonic stem cells are used along with CRISPR-Cas9 in a 3D salmon fiber gel culture system, then the dystrophin protein expression will increase in transplanted mice. The overall research objective was to remove the mutation that causes DMD and therefore increase the levels of dystrophin expression in all types of muscle tissue in a way that is efficient and can be repeated. The methods for this experiment began by collecting skeletal muscle tissue from mice, which was then grown and expanded in a petri dish. Once the cell population was ready, CRISPR-Cas9 edited the DNA found in the cells. These new recombinant cells were then grown and inserted into the experimental mice. The resulting levels of dystrophin protein expression were then analyzed at the end. The muscle stem cells that were treated through the use of CRISPR-Cas9 and a specific type of guide RNA resulted in increased levels of infection efficiency. Overall, the dystrophin expression increased in the skeletal muscles of the experimental mice. The DMD mutation was corrected in about 6% of infected muscle stem cells. This doesn't sound too high, but the figures in the next couple of slides will provide a clearer picture. The results are pretty impressive. As you can see in figure 6a, the dystrophin expression was successfully restored in the skeletal muscles of treated mice versus the untreated mice. The dystrophin is basically non-existent here in the top row, and there are many more bright patches of dystrophin shown in the green in the bottom row. Here's figure 6b. This is more of a quantitative look at the results, unlike the last slide. Dystrophin expression significantly increased in the skeletal muscles of the treated mice versus the control mice. The data supported the hypothesis and the prediction, which shows yet again how CRISPR-Cas9 can be used to alter gene expression. This gene editing tool can be used in so many creative ways, and I don't know about you, but I think that's pretty amazing. The results showed that CRISPR-Cas9 can significantly increase the expression of the dystrophin muscle fiber protein. In conclusion, utilizing CRISPR-Cas9 with a specific type of gel culture system increased dystrophin protein expression and muscle regeneration in transplanted mice. However, this method should be further replicated in order to determine what is needed to increase the efficiency before moving on to clinical trials. There are so many different avenues that can be taken from this point on to answer the following questions, like, what is the best type of cell to use to correct or prevent DMD? This experiment used adult muscle stem cells, but there are other types of stem cells that can be used too, such as embryonic stem cells, induced pluripotent stem cells, and germline cells. Other questions include which doses and what amount of time will result in the most efficient level of muscle stem cell regeneration, and in what ways will a person's immune system respond to this kind of treatment. This was a very interesting article to read because restoring the level of dystrophin expression in skeletal, cardiac, and respiratory muscles can ultimately reduce muscle weakness and help those affected by DMD to live a longer life. This is the reference for the article, and thank you so much for taking the time to listen.